Bexion's BXQ-350 is a new drug for cancer. It is an entirely brand new biologically based drug that works on an entirely brand new target on cancer. First, we will discuss the target, and secondly, we will discuss the drug itself. To understand what the target is for BXQ-350, we need to compare and contrast the biology of normal cells with the biology of cancer cells. This may seem obvious, but it's worth saying. When we have a target, knowing what we are not aiming for is just as important as what we are aiming for. The area we are not targeting is the external environment, the outside of normal cells. And conversely, what we are targeting is the external environment of cancer cells. If we could pick a one-word theme for the external environment of normal cells, it would be cooperative. And if there's a one-word theme for the external environment of cancer cells, they are competitive. So what makes the outside of normal cells so cooperative? A human body is made up of trillions of cells. They make up 13 major organ systems, and 7,500 of the parts of the body have been named. In order to build up such complexity and specialization, there needs to be a common standardized environment between and among the cells for cooperation to work. Imagine a scenario with two different cell types meeting that have two different environments. If each had their own particular pH level, both cell types would have to, number one, sense the pH of the other cell type, and number two, adjust and match the pHs in order to interact and survive. This is not a workable solution. For that reason, one standard environment and one standard pH is needed for cooperation with complex multicellular organisms. And of all the different pHs, a neutral pH makes the most sense. It's much easier chemically and biochemically to equally balance acids and bases. Keeping a neutral pH is critical for the human body. We use not one, but two major organs to do it. The first organ is the kidneys, which exchange acids and bases in the liquid form, and the lungs do the same in the gaseous form. This keeps the pH in a very narrow range, never varying far from pH 7. As stated before, cooperative normal cells have a neutral external environment. Also, the external environment does not have any destructive elements like enzymes, and normal cells do not have, nor do they need a buffer. They have a neutral lipid called phosphatidylcholine on the outer layer of the cell membrane. This gives normal cells a neutral outer shell to match the neutral outer environment. So what makes the outside of cancer cells so competitive? To answer that question, first we need to see how and why a damaged cooperative normal cell turns into a competitive cancer cell. When a cell is damaged, cancer is not the only option. In fact, 3 survival reactions are possible. It saves itself, it sacrifices itself, or it becomes selfish. When a cell has limited damage, it has the ability to fix itself. The cell saves itself and protects the tissue around it. When a cell has undergone extensive damage and cannot fix itself, normal cells have a process that recognizes this, and it will sacrifice itself in a regulated and controlled manner called apoptosis. The tissue surrounding this cell is protected. The orderly cell death process occurs as follows. Normally, cells have many highly regulated feedback loops with all of its biochemical reactions. Everything is kept equally in balance, as you can see with these reactions. Arrows pointing in both directions represents a balance with the substrates and products. Any increase or decrease with A, B, ceramide, X, Y, or Z will result in an equal increase or decrease. This is a tightly regulated system. When a cell has too much damage and is not fixed, the cell suicide process starts. A component called ceramide is ramped up to a very high level. The high amounts of ceramide overwhelm the tightly regulated system and push the biochemical reactions to go in only one direction. This cascading chain reaction effect leads to cell death. When a normal cell has extensive damage and it's not fixed and it does not kill itself, cancer develops. This is a selfish survival instinct from a bygone era. The cell becomes a cancer cell and the surrounding cells are not protected. Somehow, the damage turns on an instruction set that has been suppressed and hidden by the normal cell's DNA. It's like booting an ancient operating system on the new computer. The abilities of the normal cell still exist, but how it's programmed is totally changed. The ancient operating system comes from an ancient time before plants or people even existed. It was a time with no multicellular organisms, only single cells. It was survival of the fittest, and therefore all cells at the time were competitive. One competitive strategy ancient cells probably employed to compete was to make many copies of themselves as fast as possible to crowd out the competition. Safety in numbers, which provided survival. Another competitive strategy these ancient cells employed was to create a toxic environment to promote its own survival at the expense of others. Cancer cells booted up with this ancient operating system employed the exact same strategies. It's well known that they multiply quickly and have safety in numbers. They employ at least 3 processes that produce a toxic environment that they thrive in while causing destruction to other cells around them. Number 1, they produce an acidic external environment. Number 2, they leak enzymes from the lysosome to the external environment. The lysosome is an internal cellular component that contains acid and enzymes. One purpose of the lysosome is to recycle worn-out proteins, sugars, and lipids from all over the cell. These items make their way into the lysosome, and the acid and enzymes inside the lysosome breaks them down into smaller pieces that can then be reused in the cell. Number 3, they protect themselves from the acid and enzymes with a buffer on the outer layer of the cell membrane. The buffer is composed of many copies of a negatively charged lipid called phosphatidylserine that protects against the positively charged acid. Once cancer develops, it grows, replicates, and expands against the normal cells. It's a clash of cultures from across the ages, and it's just not a fair fight. It's ancient cells versus modern cells. It's competitive cells versus cooperative cells. It's the toxic, acidic environment versus a neutral environment. It's cancer cells versus normal cells. The cancer cells will produce their cloud of acid and enzymes. This kills off any normal cells nearby path for the fast-growing and multiplying cancer cells. We can hypothesize that cancers have at least 2 competitive survival strategies that are fundamental to its nature. 1, they like safety in numbers, so they will replicate at a very high rate. 2, they like to create a toxic environment where it can thrive at the expense of surrounding cells. Since these are fundamental survival strategies, we can say these natures are 1, common to all cancers, and 2, very unlikely to change, a conserved feature. To review, we know what the target is not with cooperative normal cells. The target is not a neutral external environment that also lacks enzymes. It is not normal cells that have a neutral outer layer composed of phosphatidylcholine. As we will go into detail later, the target for Bexion's BXQ-350 is the external environment created by competitive cancer cells, all of it. It is the acid in the external environment. It is the enzymes. It is the buffer made up of the negatively charged phosphatidylserine. Before we discuss what BXQ-350 is and how it works on such an unusual target, it's worth looking at and comparing 2 current classes of cancer drugs and their targets. Chemotherapy drugs are small, chemically based molecules. They travel through arteries and veins. They diffuse through the vessel walls. They reach into all the tissues. They are small enough to diffuse through a cell membrane into the cells. Chemotherapy drugs are engineered by people. Chemotherapy drugs block a single target inside a cell and inhibit a single biochemical process. They inhibit DNA, microtubules, the support structure of cells, and other internal cellular parts used during cell replication. Its strategy is to attack cancer's survival strategy of safety in numbers. Therefore, it can attack all cancer cell types. Antibody drugs are large biologically based molecules. They travel through vessels and diffuse through the vessel walls. They reach all tissues. They are not small enough to diffuse through the cell membrane into cells. They target markers on the surface of cells. We have taken advantage of the functionality that already exists with antibodies. The engineering of the antibody comes from the body and has been modified by people. The antibody is an excellent choice to develop as a drug. It consists of 2 distinct parts that employ 2 different biochemical processes. The targeting portion, the first part, can be modified to attach to almost anything. After the antibody attaches to its target, the second part of the antibody will trigger the immune system, which will then destroy whatever the antibody is attached to. For example, one antibody cancer drug we use targets a cell surface protein that promotes new vessel growth. This drug attempts to cut off the supply chain for cancer. As you can see, neither chemotherapy nor antibody drugs would work on cancer's toxic environment of acid, buffer, and enzymes. Chemotherapy drugs and antibodies aim at only one target, not an entire environment. So what could possibly work in such an environment? Most drugs and cells do not do well in a high acid and high enzyme environment. The answer comes from this comparison. The toxic environment of cancer cells is almost exactly the same as the inside of a lysosome. Both have high levels of acid. Both have lysosomal enzymes. Both buffer with negatively charged phosphatidylserine. BXQ-350 is a biological drug of two connected parts, phosphatidylserine, a lipid, and sapacin C, a protein. BXQ-350 can form vesicles, and that is a very important function, but the key to how BXQ-350 works is with sapacin C. Sapacin C is something of a Swiss Army knife. It's already been engineered by the body to perform at least 4 biochemical processes and normally exists inside the lysosome. Unlike other proteins, it has absolutely no problem with the high acid and high lysosomal enzymes that exist in the lysosome, and it should not have any problem with the high acid and lysosomal enzymes that exist with the external part of cancer cells. In fact, not only does sapacin C live and thrive inside the lysosome, its functionality depends on the acid, enzymes, and buffer that exist inside the lysosome. Here are the 4 biochemical processes that sapacin C does. 1. It becomes activated by acid. 2. Once activated, it attaches to the buffer lipid phosphatidylserine. 3. Once attached, it can induce fusion of membranes between 2 vesicles. 4. Once the membranes fuse, sapacin C can attach to many different types of lysosomal enzymes and catalyze their reactions. This is what sapacin C does every day in every single cell in every single person on Earth. It is what it is designed to do. It is what it is engineered to do. Let's take a look at one example. Sapacin C is able to recycle one type of lipid called a sphingoglycolipid. Sphingoglycolipids usually float on top of the cell membrane. It gets recycled and broken down by sapacin C with the following 4 steps. 1. Sapacin C becomes activated by the acid inside the lysosome. 2. Once activated, sapacin C attaches itself to the buffer lipid phosphatidylserine. 3. Once it's attached to inside the membrane of the lysosome, it is able to induce fusion between the lysosome and the vesicle containing the sphingoglycolipid. 4. Once the sphingoglycolipid is inside the lysosome, the combined sapacin C and phosphatidylserine complex can attach to many different types of lysosomal enzymes. The enzymes become engaged, and the sphingoglycolipids are converted into ceramide and glucose. So what happens when BXQ-350 is given to normal cells and cancer cells? With normal cells, there is no acid for BXQ-350's sapacin C to become activated. 2. The buffer lipid phosphatidylserine is not available for BXQ-350's sapacin C to attach to. 3. No membrane fusion occurs. And 4. It does not create any ceramide. With cancer cells, BXQ-350 does the following. 1. BXQ-350's sapacin C becomes activated by the acid in the external environment. 2. BXQ-350's sapacin C attaches itself to the buffer lipid phosphatidylserine. 3. Once it's attached, it is able to induce a fusion between the BXQ-350 sapacin C phosphatidylserine vesicle and the outer membrane of the cancer cell. 4. Sapacin C can attach to many different types of enzymes. In fact, cell membranes contain a substrate and enzyme combination that BXQ-350's sapsin C can attach to and make ceramide. And 5, the final and most important step, if enough drugs are delivered, the ceramide will be pushed to very high levels. Once this happens, a cascading chain reaction effect occurs with a series of enzymes that leads to death of cancer cells. Compared with chemotherapy and antibodies, BXQ-350 is a large biological drug. It's engineered by the body and modified by people. It uses 4 biochemical processes that are specific to lysosomes and the toxic environment created by cancer cells. Its strategy is to attack cancer's competitive survival strategy of a toxic environment which all cancers make. Therefore, it should work on all cancer cell types.